Today we are going to talk about binding between small molecules and macromolecules, proteins basically. And also this lecture will give you the basic information about protein-protein binding and interactions also. Before starting with the protein and ligand, small ligand interactions, we must first look into some phenomena, some chemical phenomena which had been discovered in the 19th century by this gentleman, Louis Pasteur. And he realized that some molecules, even though they have an identical chemical structure or chemical composition, they have a major difference. They are mirror images of each other. So these are called isomers. And isomers are kind of important for biology because, to give some examples, some drugs which have been used to treat some certain conditions has two isomers, and one of them can cause severe disorders. For example, a good example is a thalidomide. Thalidomide is a compound which was used in pregnant women in 1950s and 60s, and it caused a severe birth defect, which is called phocomelia. As you can see here, the arms and legs of the infant did not develop in utero. Actually, the chemical which is responsible for this effect is one of the stereoisomers of thalidomide. And the other isomer is quite harmless. There is also another example, quite demonstrative example, is the carvone. It is a plant-derived molecule which has a stereoisomer, and they look exactly the same, but they are mirror images of each other. And one carvone, the left-handed, so-called left-handed carvone, is actually the odor of spearmint, which gives the this characteristic odor to the spearmint. But the right-handed one is caraway odor, which is completely different than the spearmint odor. So it's clear that receptors in the nose can distinguish between these two molecules and give completely different responses to these two chemicals. The stereoisomerism occurs if the four binding sites of the carbon is occupied by four different groups. These groups can be anything, but if all four of them are different, then this carbon is called a chiral carbon, and this molecule has a stereoisomer, which means that if you have a mirror image of this molecule, they will not be exactly the same in 3 dimensions. Even though the molecular formula of the molecule is exactly the same, in 3-dimensional these two mirror images will not fit onto each other. Why this property requires a tetrahedral structure? As we talked in the previous lectures, the 4 covalent bonds of a carbon forms a tetrahedral structure in 3 dimensions. If it was not a tridimensional structure, it was a planar structure as depicted here or as shown in the formula above, then the mirror image of this picture can be superimposed on itself by just flipping around, as you can see. However, if you look at the 3-dimensional structure of a molecule, actually, which is a similar molecule with the previous one. There is an NH2 group. N is in blue, carbons are in green, and oxygen is in red, and hydrogen is in white. So this is exactly the same molecule here. The R group is a CH3 group. Okay, the R group here, shown here, is a CH3 group. But this molecule has a three-dimensional structure, and the carbons, the covalent bonds of the carbons, form a tetrahedral structure in the three-dimensional space. So when you look at these two things are mirror images of each other. Exactly mirror images. So if you try to turn one of them and try to fit it onto the second one, you'll find out that you can match three groups. You can put three groups on top of each other, but the fourth group will always stick the other way, the other side. For example, say you flip in the vertical axis, the z-axis, around the z-axis, the molecule on the right-hand side, and you flip it so that the NH2 group is on top of the NH3 group of the molecule on the left-hand side. The carboxylic acid is on the top of the carboxylic acid on the left-hand side, and the CH3 group is also on top of the CH3 group. But then you'll find out that the hydrogen will be not on top of the hydrogen group on the left-hand side, but it should be towards into the plane of the screen on the other side of the molecule. There's no way you can match these two molecules by turning them and rotating them and aligning them in three-dimensional space. These are two different molecules, but they look exactly the same. So this is an important property we will see because, for example, for amino acids, you only find one of these types of amino acids in the human or in the living organism. You don't see the other one. And this is true for many stereoisomers. You only see one of the stereoisomers. And also, stereoisomers of some chemicals and some drugs have different effects may have different effects on the organism. So there is something about the stereoisomerism, and it has to do with the binding of small molecules onto the proteins. If you remember, we talked about the three-dimensional structure of the proteins, and we said that proteins are actually solid structures, so which means that they have a surface. And also, there are certain parts of the surface which small molecules can bind with strong chemical bonds, and this binding is specific. Not all molecules can bind to these specific sites, and this has important implications for the function of the protein. These sites usually called binding sites or binding surfaces. For example, here this is an enzyme, and the substrate for this enzyme is this glucose molecule, as shown in red. So the binding site, the binding pocket, you can see, is on the surface of the protein, and glucose binds to that specific part of the protein. It does not bind just anywhere on the protein. It binds to a specific part, and this binding results in the metabolism of the glucose substrate. So for this binding, for this enzyme to function, first it requires to bind its substrate, and this binding happens in a specific area which is called the binding pocket or binding surface. It's also true for the immunoglobulin G to bind the antigen as you know, certain antigens, certain antibodies, IgGs, can bind certain antigens. So they are antigen specific. They don't bind anything, just any protein, but some proteins. They bind with a high affinity. They form strong bonds, and but they also bind to these proteins selectively. So that's one of the mechanisms how the organism differentiate between the host and the form. The general structure of the IgG is like a Y, and it has light chains, and as you know, it's heavy chains. And at the end of the light chains, as shown here, there are variable domains, which is different in every IgG, and the structure of this, the shape of this variable domains actually determines this which antigen the IgG will bind to. So, for example, in this antigen-antibody reactions, binding is also happens on a surface, binding surface, which has a specific shape, which recognize certain antigens, not any antigen. It does not bind to any antigen. It selects and binds to a group of antigens. So, what is under this specificity? First of all, we have to understand that these surfaces are not, these binding surfaces are not neutral surfaces. Here you see a three-dimensional model of a drug, which is designed to lower the cholesterol levels. And there's a target enzyme for this drug, which the enzyme which is responsible for the synthesis of cholesterol. And the idea of this drug is to bind to the catalytic surface of the enzyme, the place where the substrate is bound and metabolized or changed into products, into chemicals which lead to cholesterol. So this molecule binds to that surface and prohibits or stop the original substrate to be bound onto the protein, onto the enzyme, which decreases the activity of the enzyme, as you can imagine. In a sense, it blocks the substrate to reach this binding pocket. To do so, it has to fit into the pocket and also should form strong bonds with the side chains that make up the pocket. So, as I said, the pocket is not a neutral surface. It is formed by a lot of side chains of the amino acids, which can potentially form hydrogen bonds. And other bonds also, but mostly hydrogen bonds. The hydrogen bonds are important type of bonds that play a role in these type of reactions. So, first of all, the molecule should be able to fit into the binding pocket, and its shape is important so that the parts of the drug or the parts of the small molecule should be able to form hydrogen bonds with the appropriate side chain groups that is protruding into the binding pocket. Here you can see the three-dimensional structure of the drug and also the place where it binds to which fits into and binds on the surface of the protein. The protein is shown as a yellow surface. All those things you see are chemical groups on the side chains. On the right-hand side, you see a schematic representation of the same drug in black. And also the hydrogen bonds that are formed between the small molecule and the side groups of the of the amino acids on the surface of the protein. They're also shown in yellow color. You see that it's not only the shape of the molecule, but also the molecule has the appropriate hydrogen bond donors or acceptors in the right place, so it can form all these hydrogen bonds, which makes the molecule quite stable in that place. So once it gets bound, it does not come apart easily. And also the molecule has to have the structure so that the enzyme won't be able to catalyze it, change it, so the drug won't be metabolized. So this is a very good way to understand the small molecule and protein interactions, where the specificity comes from. Why handedness is important, because this binding into this pocket can be seen as a fit between a glove and a hand. So if you try to fit a right hand glove to a left hand, you'll see that it doesn't really fit very easily, even though the shape of the left glove and the right hand glove is quite similar. A left hand glove won't fit into a right hand. So it's like here the stereoisomer of a drug won't be able to, even if it looks very much like the right-hand drug, the left-hand drug won't be able to fit in here, or if the left-hand drug can fit here, the right-hand drug won't be able to fit in here. So the stereoisomers show quite different activity, may show quite different activity in the organism. And also during the synthesis, this binding pocket determines whether the left-hand or the right-hand version of the molecule will be synthesized. That's why you see only one stereoisomer of a substance, for example, an amino acid, is being synthesized by the organism. So there's an important definition here has to be made. There are two types of binding reaction between a small molecule and a protein. This can be illustrated with enzyme inhibition. There are two types of enzyme inhibitors. One of them is called the steric inhibitors or blockers, or allosteric inhibitors. We've seen that in order to an enzyme to function, the substrate had to bind to a certain part of the surface, which is called the catalytic size or the catalytic pocket. And once the substrate binds there, then it gets metabolized and changed, and the enzyme functions. And you can synthesize, as we have seen in the previous slide, a pseudo-substrate that binds into the pocket, fits into the pocket, binds to the pocket strongly, but inhibits the real substrate to bind, and in a sense it blocks it. It competes with it and it blocks it. The pseudo-substrate cannot be metabolized, so the enzyme stops functioning. This is steric inhibition, which happens on the same binding pocket. The substrate and the pseudo-substrate binds to the same surface. They may not form similar, exactly the same type of hydrogen bonds, but doesn't matter. The pseudo-substrates also fit into the same binding pocket as the substrate, and in this way stops the function of the enzyme. In the allosteric inhibition, on the other hand, there are two sites on the molecule. One is the catalytic site, as we discussed, and there is another site called the regulatory site. And if a substrate or if a molecule that can bind to the regulatory site causes a conformational change in the molecule of the enzyme, the protein, which in turn changes the behavior of the protein, and either it reduces the binding of the substrate to its catalytic site, or the catalytic action of the protein is reduced. This does not occur, this interaction does not occur on the same binding site. There are two binding sites. One binding site through the protein affects the function or the properties of the second binding site. This is called allosteric inhibition. There are two. The interaction is happening at a distance on the same molecule, but from one binding site to the other binding site, there is an information that flows and affects the other binding site. Many proteins can be regulated. Their activities can be changed by this mechanism of allosteric inhibition. Okay, so this is the end of the series of lectures. I try to give you a general view of the protein structure and function. See you next time.